Hello and welcome. I'm Luciano Lucas. It's a pleasure to be here and represent the Aivia team today. I will be introducing the new functionality of Aivia 9.5. Today's agenda will include a brief introduction to Aivia tools and also the roadmap that we've been following and some hints about what we're doing in the future. Then I'll do a very brief introduction to the 9.5 features. This is followed by several demos, and then there will be some concluding remarks and time for questions and answers. The Aivia product family and friends has been growing over the last 3.5 years. We have Aivia itself, which is a desktop application. We have Aivia Cloud, which can be ran locally, but also connects to GCP, so the Google Cloud platform, and allows you to train and apply deep learning models using cloud computing. We have also Aivia Web, which we officially launched earlier this year, and it is just like Aivia in terms of functionality, but instead of installing it on your local computer, you run it via a web browser which accesses a cloud computer. And Aivia Community, which we launched at the very beginning of this year. This is a freeware, essentially a viewer, but also allows you to create beautiful video animations as well as collaboratively create annotations to train machine and deep learning models. Over this year, we've also added a few friends. First of all, our GitHub repository has quite a few Python plugins there, as well as the models that we tested for our preprint, the 3D Arcan preprint, which will be mentioned later on today. There's quite a lot of goodies in there. Our Aivia forum, which has become a good way to get support and to provide support as well to our users. And the Aivia Deep Learning Model Library, which will be introduced today. It's hosted on our website and it'll include several of our deep learning models. Over the last 3.5 years, we have introduced quite a lot of new functionality, and we focused both on the AI microscopy side of things with tools for classification of objects, detection of objects, and also image restoration. But also a lot of image analysis functionality for neuron tracing, object detection, and virtual and VR image exploration, and much more. This is the full list. I won't go into the full details, but I just wanted to highlight that we are pioneering technology in the AI microscopy space ever since we started with Aivia. So in the beginning of 2017, we were already releasing functionality. So if I pull out just the machine and deep learning functionality, this is what it looks like. So we've been very busy throughout not only developing standard image analysis functionality, but in parallel adding quite a thick layer of functionality that uses a machine and deep learning to our advantage and to the advantage of all the users that have Aivia. So Aivia 9.5 has quite a lot of new functionality, both on the AI microscopy side of things, but not only. We will introduce today 20 new models that we've pre-trained and they're ready to use. They cover 7 organelles and 3 different imaging modalities. There's also a library that is online and you can use it now. I'll share the link in a moment. We have developed a couple of ways for you to deploy these models. So we will share that with you today and discuss those. Actually, there's 4 ways and I'll show those in a moment. We've also expanded our cellular analysis recipes. We launched a 3D version, and now we have a 2D version as well. And there's some additional batch functionality. There's a whole new tagging system, and this allows you to not only tag points in 3D space with free text and comments, but also search for those and then navigate to those. So the camera will actually zoom in and zoom out towards the points of interest. In terms of usability, we have increased the usability quite significantly by adding the possibility to define your own UI layout. This is pretty powerful. You can have layouts that are specific for whatever application you're interested in. There's also now the possibility to store multiple storyboards so that each image can have multiple storyboards, and therefore you can keep different stories together with your image. So important for the whole community, and also for us, for our research and for the work we're doing, is adoption of deep learning for microscopy applications. And we are doing everything we can to help with that adoption. And the first thing we do a lot of is try to provide educational resources to try and help the community also explore this option. In addition, we do a lot of R&D and we test, qualify, and quantify the performance of these models versus ground truth and in stringent scientific conditions. We then deploy in the easiest possible way the models that we train, and even deploy them on GitHub so that it's easier to test them, so the wider community can test them and explore how they behave. We do provide full technical support to the models that we deploy within Aivia. So these are professionally supported by our team, and that provides you some security. We are continuing to develop these models, and we are happy also to help you develop additional custom models. So that is something that we also provide. And as we develop new models or update the old ones, we will be replacing the old ones with the new ones, so that you can use always the best quality models available. The models, or many of the models that we are going to introduce today, were actually they are actually part of a project that we have ongoing with a large group of researchers, many of them at the NIH and Bethesda, but not only. This work was presented or introduced as a preprint, which you can explore via this address, this link here. We've also placed all the code in that. So that's what the paper looks like. We also have all the code on our GitHub, the training code and the apply code, in case you want to do that in Python. Here it is, with full descriptions and some example data, so that you can try this. The paper includes a comparison across different modalities, but also it includes analysis of how the model, our model and other models behave when restoring, doing image restoration tasks for multiple organelles. We did that using the iSim, but also using confocal to STED, and then expansion microscopy. For more details on that paper, you can visit the AI Microscopy Symposium. The first edition of that symposium was just a few weeks ago, actually just 3 weeks ago, and that symposium included presentations from the Allen Institute, also University of Washington, researchers, and the University of London, the City University of London, and myself. We covered, the presentations are actually recorded and shown at the bottom, so you can watch in some detail. My presentation will include a longer explanation of what the paper entails and showcases some of the results. So in summary, these are all of the models that we currently have. You can see there's quite a long list of them. Almost all of them are already on our online model library, and all of them will be available through Aivia itself. And we're going to continue to populate the model library online. So as you can see, it covers not only denoising, but artifact removal, super resolution, virtual staining, and segmentation. The model library looks like this. It's aviasoftware.com/model library, and each one of those tiles goes to another page where you can actually read about how the model was trained, what data was used, what kind of architecture we used, and then you can download the model, the trained one. You can also download test images, and those will run in Aivia. Generally, the training happened in this way. We have target data or ideal data, also the input data, which is low quality. We trained a deep learning model, in this case an Arcan, and then this Arcan, when provided with similar images to the input data, which are lower quality, it can restore the quality of those images back to similar quality to the ideal or target images. The deployment is done in such a way that helps you use these models in an easy way. So you can just drag and drop them into the Aivia recipe console, and that will create a recipe that you can just run. You can also run them in the batch applier in Aivia. That's a possibility. You can also run them in Aivia Cloud, both locally and using cloud computing. And you can use any of the recipes now can also accept a pre-processing step. So in this case, I'm showing the cell analysis recipe, and there is a pre-processing step here where you can just simply load the model that you want to use, and that will be the first thing that happens to the input data, to the raw data. It's processed by the deep learning model, and then it continues to do cell detection, in this case nuclei detection, vesicle detection 1, and then 2. So in the first 3 cases, we get the new channel as an output. In the last one, we actually get segmented objects. But even if you don't do the last one straight away, you can just take the new input channel, take the new channel as an input to any of the recipes or to the pixel classifier, and do analysis that way and get objects in that way. So here you can see, this is just a brief video highlighting how you can get the models in. These are the trained models. I'm just going to drag and drop all of them into the recipe console, and they all convert to recipes, and they are now added. If I select one of them, now they're ready to use. That's it. Once you apply one of these models, and in this case I'm applying a model to, I think, the 3rd channel, it will create another channel, in this case called result, and I only plotted on the small ROI. So you can see here, in this case we use the confocal to STAD model, so it increases resolution of the image. And even though the model was trained originally for nuclear pores, here it's used for protein 95. It's a postsynaptic density marker, and you can see that the quality has increased significantly from what you can see in the green to the white signal. So we'll have a look at this data in a moment. We also have virtual staining models, in fact 3 of them. And so from a bright field image and a real fluorescence image, this is a real image. Both of these are real images, actual images on a microscope. We can train a model that then can predict and virtually stain the bright field image, another new input image that is just a bright field image, to look like the one on the right. This is an actual different image. You may notice some differences between the center image and the one on the right, but the one on the center is a real image. The one on the right is a virtually stained or predicted image based on the bright field image alone. We'll have a look at this as well during the demos. For segmentation, the general training process is similar to the general training process for image restoration or virtual staining, but here, instead of introducing high quality, high signal to noise, higher resolution images as ground truth, we use manual annotations or automatic annotations, but the image needs to be annotated, and then the raw data. With these two, we can train a model. The model can then predict a new channel that is a confidence map for each of the pixels, and then from that you can threshold it and get segmentation or 2D, 3D objects. Downstream, you can further segment the image, and you can see here 2D segmentation and 3D segmentation. In this case, it's an example of an electron microscope image that was segmented using one of our pre-trained deep learning models, and these can then can create beautiful renderings, but also getting quantitative information about the numbers and the volume and the shape. In the future, we will be able to do even more. We're actively working on a project called Intelligent Connectomic Analysis tool called Leica, which you will hear more about in the near future. We've also created a new system. It's a tagging system. The new tagging system, you will find it as one of the new sub-tabs on the right-hand panel. You can see it highlighted here. It looks like this. So you can create a tag in 3D and 4D. These tags can be free text plus a comment. They can also have a specific color. Each tag can have its own color. And you can save these tags as presets in case you have a group effort and you want to make sure that everybody's sharing the same tag, so you can share the tags with them so that they are properly annotated. The images are properly annotated. This is what it looks like when you have a few tags already in use. In this case, I just have 3 different tags, but I'm using one of them 3 times in different locations on the image. So what does it look like when you're using it? This is a brief video that illustrates that. So you can also use these to search and to navigate. So in this case, I'm selecting a particular tag. They're the large N on the membrane, and I selected it, and I'm navigating to it, and I'm showing that it's actually on the membrane. And I know that because I first detected the cells, detected all these puncta, and then one of the things we can do with the 3D cell analysis is measure the distance between particular puncta and the cell membrane. So this one is considered to be large based on this scatter plot, large and is very close to the membrane, and I wanted to place a tag on it that is related to those 2 characteristics. You can set as many tags as you like, and you can always search them in the search bar inside the tagging system, and that will allow you to quickly find the tags that you already created. Aivia now also has a way to customize the UI. This is one of my favorite, very simple but very nice features. You can pin, float, dock, hide, and show the different layouts. You can find the control for this functionality at the very top right of your screen. It's a little cogwheel. If you press that, you will see, initially you will see nothing in this space here. There will be no layout presets, but you can create your own. And the next time you load Aivia, they will be there for you. And if you want to use them, just hit the little button on the left and that will change the GUI. So what does it look like when you're using it? So this is a brief video. If you click on it, it will just rearrange the whole GUI so that it is just the way you had set it up. I have several favorite layouts now, and I use them a lot. So this is some examples. This is the one I use for visualization. Everything else is hidden. The only thing I have is the volume rendering controls and all the channels. So this is the visualization one. I have another one for doing image analysis, where I have quite a large window to look at the data, but then two panels, one that controls the objects and the rendering, and the other one that controls the actual recipe that I'm about to run or have already run. Then if I've done all the analysis, I want to explore the results. And in this case, I need the spreadsheet, the charts, and the object sets so that I can easily control what is visible in the 3D view and interact with all the data. If I'm annotating, I use a different view to maximize the space for the image and keep the tools that are needed for annotation well in view of my screen. I do have multiple screens, and many of you also have multiple screens, so this is a great way to maximize on that real estate. You can spread all your controls across the two screens. That's what I've done here. So this screen one has the volume rendering and the charts, and then on the right hand side I have everything else basically ready to use. We have also expanded our 3D cell analysis or multiscale image analysis in Aivia. A few months ago I was introducing you to the 3D cell analysis, and these are some images from that launch for 9.0. These are all 3D examples, but still very relevant. The main concept here is that we move away from just analyzing the images on a per image basis, but we focus on biological elements and biological entities. And this gives us so many more possibilities in terms of extracting meaning from these images that are either labeled with multiple channels, or we can create multiple channels using the pixel classifier or one of the deep learning models. So the 2D cell analysis is very much like the 3D cell analysis. The layout is very similar. Here is an example. So the image on the right is a 2D image with 3 channels. The segmentation is what you see on the bottom. And if I peel away the cell detection itself and to show the vesicles and the nucleus, then I can color code things and use all the nice functionality that is standard in Aivia, the 3D, the interactions between the different panels and the different views of the data. So this is what it actually looks like in real time when we're using it. So previews of each of the channels or each of the biological components, and then you hit start. That will run the full pipeline and create an object set, a group, in this case cells. Or very quickly you get the measurements calculated, and you can start to interact with any of the measurements that are pre-calculated. It is possible to add a lot more measurements through the spreadsheet tab, but in this example I just have a few measurements to explore the results. We'll have a look at this in the demo in a moment. So we quickly got to the demos. We'll spend a little bit of time, first of all, looking at the actual library of deep learning models. Then we'll show how to deploy these models in Aivia. Next, we'll talk a little bit about the tagging system and show how that feels and looks like in Aivia. then the UI customization, and finally the 2D cell analysis recipe. This is the deep learning model library. This is created by Hoyen Lai, who is also a first author of the publication, the 3D Arcan preprint that we put out at the end of August. So in the library, you can access through this address. You can see that there's already several models listed. And I've been using during the presentation, and we'll use in the demos, this model, for example. So I could go in there and see the page related to that model. So it shows typical image, a little bit of a description about the source of that model, how we created it, what kind of imaging was done, requirements, and then you can download both the model, the AC model that you will see me use, a test image, and you can download the whole package that way. There's also some installation details on every one of these models, and you can download everything. For this particular model, you can download everything just hitting the download button down here. If you have a particular interest, for example, you're interested in nuclear pores, that's the one I was just looking at, you hit that and it will sort the whole gallery with that particular tag. So denoising, we have several, and if you want denoising in Golgi, then you have this one. You can go in there and read around the topic, and then download it and apply it in Aivia, as you will see in a moment. So this is a great resource. I welcome everybody to come and check it out. We will be adding more models as the days and weeks pass, and as our R&D team builds out new models, we will deploy them here, as well as directly in Aivia through the update models in here. New models will come up here. So this is for 9.0, but once we have 9.5 out, so later today, you will then be able to see all the new models listed down here. In this first demo, I'm going to illustrate how to apply a pre-trained deep learning model to a confocal image. This is the confocal image right here. It has two channels, nuclear pore complex here and the microtubules here. I'm going to just apply the confocal to STED model on the nuclear pore complexes. I also have the real STED image just for comparison, so that I can compare with the restoration once we get it. So to get the model, first of all, I need to either download it from the website, or I can get it through help, update, and then models, and download the model that I need to use. So if I download it from the website, I will have it in a folder like this, and all I have to do is pick up the model, drop it into Aivia. Aivia converts it to a recipe, and then I can apply it. I'm setting up a smaller ROI so that we can do this quickly in real time here. Then I select the channel that I want to apply it to and hit go. Just going to bring in here the task manager so you see what is happening here on my GPU. So you can see it has already happened. And now I have a result channel. This result channel has the restoration that I have created using the pre-trained deep learning model. So that's what it looks like, the confocal, and that's what it looks like for the restored image with this pre-trained deep learning model. I can go in 2D as well and make, for example, a line measurement here just to check how these peaks look like. And you can see it here. So this is the restored image, The confocal looks like this, so there is really no separation between these two peaks. With the stead, there is a clear separation, and with our result, there is also a clear separation. So clearly the resolution has improved quite dramatically using this pre-trained deep learning model that we just dropped into Aivia. Now, taking the same data set that we were working on just a moment ago, I'm going to do, first of all, the detection of 3D objects using the standard confocal image. And I already preset the parameters so that I can get the best possible partition and detection based on this channel. So I'm going to create that. That's what we see now. And even though it is a confocal image, not as good as the stead for sure, we did manage to partition quite a few of them where there is ambiguity, like here, for example. But now I will do the same thing, but instead of applying it the standard recipe, I'm going to add a pre-trained deep learning model here, and then run the same parameters, and that's going to first of all apply the confocal to stead model to the input data. Then it's going to do the detection, the partition, and it creates me these objects here. So that's, let me just relabel this DL. So this is the deep learning version, and I'm going to make these blue and the other ones yellow, and turn this off for now. And let's have a look. So definitely the yellow ones are bigger. You can see the blue ones are inside, they kind of disappear. So first of all, they're bigger, and secondly, in quite a few cases, there are cells that are not partitioned properly, or nuclear pores that are not partitioned properly. So you can look closely here, for example, where with the confocal data, even though we tried really hard, it would still detect just a single big blob. And the raw data looks like this, so it's no big surprise that a big blob is detected. With the stead data, or the deep learning restored data that aims to mimic a stead data set, we can actually partition these. And if we look at what the channel looks like, the process channel looks like, that led us to create these objects in deep learning, it's no surprise, because the data is actually pretty good now. In fact, if we worked a little harder, we could probably even separate a little these that are still kind of together, but there are two peaks here, so we might still be able to do better than this. So in conclusion, for this demo, we applied a pre-trained deep learning model as a pre-processing step in a standard Aivia recipe, and you can apply it to any of the recipes that we currently offer. All of them have this step. We then did image segmentation, and I compared that, compare the segmentation done on the raw image, which is this, so the confocal image. This is the segmentation we managed to get, and this is the segmentation we managed to get using the stead image. So quite a bit better in terms of partitioning. And size. Switching gears a bit, we're now going to use one of the virtual, or actually the 3 virtual staining pre-trained deep learning models to process this image, which is a bright field image, and create something that will look similar to this. So this is the ground truth data that we use to train the model together with the bright field. So we train the model to predict from the bright field actin, microtubules, and nuclei. And you can see I've already done it earlier. Just in case the demo doesn't go right, I can show you something I've done before. But right now I wanted to show you the process. So the process is to take each of the pre-trained models, loaded in the same way that we did before. So load it in, it converts to a recipe, and then take bright field number 1 and apply. And that's for the actin. So we're predicting actin right now. And I'll do it in this sequence. It will be actin, microtubules, and then finally nuclei. And then I'll go and rename these. Okay, so this is all running on the GPU. So it's pretty fast. And I'll label these. So this is actin. I have to just gently adjust the lookup table so that we can see this. So microtubule and nuclei. Yeah. Also change the colors to match. So that's what it looks like. It is, they tend to be brighter than the original, so I'm going to lower this down a bit. And also the nuclei has more background than the original. So that's the original. These are the nuclei that we just predicted. Then the actin original and the actin predicted, and the microtubules original and microtubules predicted. So we have done that part, which is predicting the 3 channels from just the bright field image. But let's go further, one step further, and do the analysis of these nuclei, because it is really difficult to do segmentation of a data set like this. And the use case here is all you have is a phase contrast image, and you want to keep imaging for a long time, no fluorescence imaging at all, just do bright field imaging, but then you want to easily count the nuclei. So that's what we would be able to do with this model. So here's the predicted nuclei that we created based on the phase contrast image. And now we could use the the nuclei count recipe right here. Make sure we're selecting nuclei as input, so this channel as input, and we can do the detection, the partitioning, and that's the result. So pretty good. If we wanted to, instead of doing this in 2 steps, so I did it in 2 steps. I first processed the bright field image to create the nuclei channel, and then I ran the recipe to create the objects. I could directly apply here the model that I want to apply, so the virtual staining nucleus. In this case, the input would be bright field 1. So I'm going to take this bright field 1, process it with this pre-trained model, and then do the detection. And I'm going to create a new object just to compare to this old one that we just created. And I start. So this is starting from scratch, pre-processing. So we'll see CUDA going, and then it creates the object set. So now we have 2. This is the one we just created. I'll color all of them yellow. And this is the previous one. They're identical, of course. So that's the first one we did, and then the second one we did. And they are identical. Either way, either in the 2-step manner or in the single-step kind of recipe with the pre-processing step, you get to the results, and then you can explore these results. Actually looking at these, explore these results in the charts or in the spreadsheet down here, and there's even more parameters that we can add if we like. A third way to do this is with the pixel classifier. In this case, we do pixel classifier. I'm just going to create a new one here. Make sure I'm using the nuclei as output. And I want to do smart segmentation. Okay, and then want so I'm highlighting a few cells that are somehow representative of the group of cells here. And then I can generate a preview. So that would be how I would do that. There's still some partitioning mistakes here. This can be relatively quickly fixed by giving a few more examples. Like this. Okay, and hopefully that will give us a good partition. We could continue to train, of course, to get even better partition, but this is just to illustrate how we would do this. Then teach, apply, and now we have segmentation done also for, in the case of the pixel classifier. So we did it, the first way was with 2 steps. So we processed the channel and then with the deep learning model to create the nuclei, then ran the recipe. The second way was by running a recipe with a pre-processing step embedded, and the third way was to process with the pixel classifiers. It's a machine learning segmentation approach, which is here, to process the channel that we created with a deep learning model that predicts nuclei channel from a bright field image. Briefly, I wanted to show you also the tagging system that we now have. I already loaded this image, beautiful image from Wanqing here at UW, and did some segmentation. And here is the tagging system down here. So if I have some tags, which I do, in fact, in this case, I already tagged this image with a few key things that I wanted to highlight. For example, a small and near the membrane cluster of protein 95. And so I can click on it and it will navigate to it and highlight it. And you can see it here in white. It's right there. And I know that this is a small one because I use the charts and I use the charts that I can get from here, analysis tool. So I can see that from one of the charts. So there, and distance to the edge of the cell, and also the size. Volume. So that's one of those right there. So it's relatively close and it's relatively small. I also tagged some large ones. So here's one large one right there. And this one is, if I select it, it will be highlighted up there. I can also color code these for distance. So it's a little bit easier to spot, and you get the idea. So I can tag these. These are tags that I already made. You can also create preset tags. That's what I have here. So if I wanted to share some work with a colleague or a student, a collaborator, I could have a set of preset tags like this, which I can add more to. I'm just going to add one now for testing. So test. Can choose another color and add that. So that adds it here, and I can then export these presets and give it to, potentially give it to a colleague, and he can load it into his Aivia and get these same presets that I have just created now. To get them back in, it's very straightforward. So if I delete this, there's I want to delete all of them actually. Okay, so I deleted all of them, and then I want to import. There's my presets, and I get them back. So I have the presets right here, and if I wanted to highlight a few cells that I need somebody to check for me, or a few clusters, I could do it this way. And that will tag all of them. So you can see here, maybe I would finish up reviewing some others in a different location. So I can use a different tag, for example, done, if I was convinced that I had checked. So that's done, that one is done, and that one is done. So that basically allows me to tag and navigate, and then I can search. So if I want to just see the ones that are done, I can quickly get to them and see them. Right now they're red and they're kind of behind, but you can also make this transparent, and that becomes a little bit more apparent where the tags actually are. There they are. So that is, in a nutshell, the tagging system. Following on from the work we did for Aivia 9.0, we've actually created now the recipe for cell analysis for 2D. We have the 3D cell analysis up here and the 2D cell analysis down here. So this is a data set done on a Sora system from 3i. There's 3 channels. They're beautifully imaged nuclei, looks like microtubules, and then some proteins that are labeled here in this blue channel. We can do the detection. As before, you have inputs. You can have multiple vesicle types, up to 5. I just have 1 selected here. And then you do the normal process. So this is a preview. There's the parameters, preview of the cell, preview of the nuclei, and preview of the vesicles or clusters of proteins, and then you can apply it. You can see I already applied it before, just in case something went wrong in the demo, but I will apply it now so you get a sense of how long it takes. So that is now complete, very, very fast. And I'm going to color code the cells by cell, so each gets a color, and I'm going to turn off the cells so you can see. So there's a few cells there, and as usual, you get all the measurements related to each of the cells. So this is the nuclear I actually wanted to, yeah, nuclear can also do. So that's contain number of vesicles. That's an interesting one. So some have more, some have less. Okay, so that one clearly has more, the other ones have less. And then the vesicles, you can also color code them by, for example, distance to the center of the cell. You can see that there's the cell right there. And that's distance to the center of the cell. All the measurements are down in the spreadsheet as well as with the previous ones. And to access the different parts of the cell, you have the drop down on the left here. I wanted to thank and acknowledge all our collaborators that help us explore new avenues of development for Aivia, and help us also ensure that what we develop is of the highest quality and can be used by the wider microscopy and cell biology community. In particular, in this release, the main contributors were certainly Harry Sharoff and Gigi Chen and their respective teams, as well as Chris Combs and his team. Primarily they, but a big thanks to all of our other collaborators as well. We do appreciate all your input. Also, a huge thank you to my team. Outstanding effort to get Aivia 9.5 released on time and with so many new features that will help so many of our users do their research and advance their science. It isn't an easy time for a lot of us. Everybody working from home, some people with their children at home, others with elders to take care of. So this is a particularly challenging time for a lot of people, but still we continue to create good tools, and we're happy to share them with you. And I'm very happy for my team, for their achievements. So I wanted to acknowledge a couple of people, or a few people in particular. Hideki, who is one of the first authors on the 3D Arcan paper. Hoyen Lai, who is another first author on that paper. Sean, who has developed the tagging system and he's working on really cool future tools that will come online very soon. So is Bryce, has been very busy and of course helped a lot in this release as well, helped the rest of the team. Casey not only helped develop or actually developed a lot of the implementations for deploying the deep learning models in the recipe console, also worked on the 2D cell analysis. Chris, who is our image data handling guru, thank you very much for your efforts in this release. John, who has been instrumental with Aivia Cloud, but not only, and has done a lot for the deployment of the Aivia models that you'll be using now. Michael and Aiden are our test and QA team. They are outstanding. They keep going and keep finding bugs and finding problems which we then can fix before the software gets to your hand. So absolutely essential for us to have them on the team, and they're doing a great job. Hanyu, who started this year actually during the pandemic already, has had a great input into multiple releases now, and in this one he trained several of the new deep learning models, and he continues to have a great contribution. Ijung, Pluto, is another one of the first authors of the 3D Arcan paper, and he created a lot of the data, actually the raw data that we use to train the models. And finally, I wanted to acknowledge Chi, who is in fact our algorithm wizard. He collaborates with everybody on the Aivia team and helps his sub-team, the algorithm team, to develop the algorithms that we need for all the recipes, but also the deep learning models. So in conclusion, I wanted to say that now Aivia has the largest pool of systematically characterized and commercially supported deep learning models. And this is really an outstanding achievement. We are, as you know, a relatively small company. We've done a lot over the last few years, and now we have 20 models that are already out there, or will be now from today. And we have several more that we are working on and will continue to develop, qualify, quantify, characterize, and then release to you together with example images so you can test them out. You can now upgrade your favorite image analysis pipeline with a pre-processing step that uses one of our deep learning models. This can be extremely powerful, as you saw in the presentation today. We do have probably the easiest way to deploy deep learning models I know of. It's a simple drag and drop. It cannot be much easier than this. The UI is super flexible. This is also should be really a pleasure to use for everybody. It is certainly for me. And I didn't show this too much, but the storyboard, having the possibility to save multiple stories within that storyboard is really great when you're preparing to give your presentation or doing your lab meeting. And together with the tagging tool is really great to tell your story in detail. So with that, I want to close and go to questions, and hopefully I'll give some answers as well. I'd like to invite you also to join the AI Microscopy Symposium. We have one that happened a few weeks ago. The next one will be in December, beginning of December. There is an end of 2020 promotion in case you're looking to purchase a license of Aivia or Aivia Web. There are promotions that you can have access to. So contact your local Aivia representative. And I do encourage you to try Aivia and Aivia Community and Aivia Web, all of it, as soon as possible. You can download them now. You can use Aivia Web right now. Just go on the website and request a demo, and you'll be sent a link that you can immediately use. With that, I thank you for joining and open up the floor for questions.